Hi, I'm Mr. Baker, and these are your notes for 7.2, Meiosis I and Meiosis II. You can see on the opening slide we have our silly word to remember the phases of Meiosis I and II, and it's ip-mat-C, p-mat-C. You need to be aware of homologous chromosomes. And homologous chromosomes are ones that have been doubled, which has two from mom and two from dad. They match. That's the big thing. They match. Review of haploid and diploid and gametes. Gametes are the sex cells, either the egg or the sperm. They have half the number of chromosomes as the rest of the somatic or body cells. In humans, that's 23 individual chromatids, not pairs of chromosomes, in the egg or sperm, depending. They're haploid. Haploid is represented by the little letter N, found in the sex cells or gametes. And for us, N equals the number of sister chromatids. Diploids are represented by 2N, and that's the somatic cells, which is any other cell in the body but the gametes. Don't forget the silly word to remember the phases, ip-mat-C, p-mat-C. Meiosis I is ip-mat-C, Meiosis II is p-mat-C. Notice that the I is missing the second time through, so there is no second interphase. Meiosis I starts with interphase, and it has G1 and SG2, which you should remember from last chapter. And prophase 1 has a couple of special things that appear. One of them is that they form tetrads. Tetrads are 4 chromatids that are stuck together. And the way these are interesting is because when they form together, they can trade pieces of DNA with each other, and that's called crossing over. Centrioles move to opposite sides of the nucleus, the spindle hooks the chromosomes, and then the aforementioned crossing over occurs. Well, what is crossing over? It's when you trade pieces with each other. You can see the orangish and the dark red, and then you have a piece that's an orange one that's got some dark red, and a dark red that's got some orange. Crossing over, it's a trade. Metaphase 1, we're going to line up something in the middle whenever you see metaphase. Anaphase, we're going to split or rip some chromatids apart during any anaphase. Telophase 1 is going to get 2 nuclei, and cytokinesis 1 is when we get 2 daughter cells. That takes us to meiosis II. Remember, no I, it's p-mat-C, so there's no interphase. Go directly to prophase 2, where the chromosomes appear, centrioles move to opposite sides, and the spindles hook the chromosomes. Metaphase, they're going to line up in the middle. Anaphase, you're going to rip those chromatids apart again, or you're going to split them apart, whichever way you'd like to remember it. Telophase, we're going to form 2 new nuclei. We're going to have cytokinesis 2, where we're actually going to then result in 4 haploid daughter cells. That's for if you're producing sperms for males. Females do it something slightly different. There's a couple pictures to help you visualize, and the number of chromosomes and the count. Again, the whole point of meiosis is to increase genetic variation, because how the chromosomes line up and get split apart is completely random. You're shuffling the genetic deck. And crossing over is further going to increase that variation because you're going to trade pieces of cards following the analogy of shuffling the deck. So it makes it even more different. Here is a table. You'll need to memorize that. You may get a quiz where you have to fill in the blanks or the words that we've removed. So here is mitosis. 1 cell doubles, splits into 2. Meiosis, 1 cell doubles, splits into 2. Doesn't double, splits into 2 again. So you cut the number of chromosomes by half, and you end up with N as opposed to 2N. Genes are typically found on different chromosomes. However, genes that are found on the same chromosome are called linked genes. All you need to do here is memorize. Linked genes can also increase their variation by trading pieces back and forth. So here we've got linked genes, a blue and what appears to be red. The blue and the red will switch a little piece there, and so it changes them up and gives you another chance for variation. That's what I want you to know. A phenomenon that is not necessarily good is called non-disjunction. And I will argue that's a kind of a dumb word because it literally translates to do not join. Or we could just say stick together. You have to remember non-disjunction. What happens during non-disjunctions, the sister chromatids don't split during either anaphase 1 or anaphase 2s, which means you don't split them up. Everybody gets a fair shake. Somebody is going to get an extra chromosome. Somebody's not going to get one. They're going to be missing a chromosome. What does that look like? Well, let's take a look at a karyotype, a picture of chromosomes. Here's one. This represents Down syndrome. And look down here at trisomy 21. That's chromosome pair 21, which should only have 2, like all of the other chromosomes, but it actually has 3. Are you looking at 21? Because it's on fire. Yeah, hard to miss that one now. Also, this person is a female because she has XX on the bottom right. Sex chromosome comes in 2 flavors in humans. In females, the X chromosome is the female chromosome, and the Y chromosome is the male chromosome. To be female, you just don't have to have a Y. XX is female. XY is male. You can have too many or too few chromosomes. The syndromes I expect you to know are Turner's. X and the 0 because it's a missing chromosome. It's a female, instead of having 2 X's, only has 1 X. And those females, you can read up here, short height, web of skin between neck and shoulders, underdeveloped ovaries. They often are unable to produce children, and they might have some learning difficulties. Klinefelter syndrome is XXY, which is where when a male gets an extra X. And this might be underdeveloped. These symptoms can occur: underdeveloped testes, lower testosterone levels, usually infertile, meaning can't produce viable sperm to have children of their own. Might have development of some minor secondary female features, and long limbs and slightly taller than average. Then they've got listed here something called a supermale, which is not a big deal to us, but it's XYY, where a male gets an extra Y chromosome. You can see there are some implications for that. And no, they're not actually superhuman or they do anything incredible. Usually, if an individual can survive with an extra chromosome, there's going to be something not as good as normal. And then there's something about this metafemale, but I will argue that this metafemale thing is false. It's also called trisomy X. This is all optional, this slide. And it's typically not a thing. Why? Because interestingly enough, females will shut off the extra X chromosome, which is kind of interesting. Monosomy, which is Turner syndrome, meaning there's only 1 X chromosome instead of 2. It's missing there. They've got it shaded in the bottom right. Klinefelter's is XXY. There's your normal karyotype, and then you got the X, 2 X's and a Y. And that is not normal. It's Klinefelter's. You'll need to know those for your quiz. And speaking of your quiz, these are topics that are fair game. The homologous chromosomes, haploid, diploid, IPMAT-C, PMAT-C in order and what occurs. You'd better know when crossing over occurs, the differences between Meiosis I and Meiosis II. Don't forget the silly word, or your silly phrase, IPMAT-C, PMAT-C, the definition of linked genes, the non-disjunction, and the 3 disorders that I covered in the notes. Study hard, and we'll see you on your quiz.